and we'll be done. And then I'm going to give you the assignment. You can just work on the assignment. Okay, so we began our journey into the mollusks and talked about 4 classes that your book mentions. Just know that there are more, but these are the 4 that you need to be aware of. The Polyplacophora, which is many plates, 8 plates kind of fused together. Gastropoda, that's our terrestrial class. The bivalves, 2 shells, and then our cephalopods, which is their squid, octopi, and that's where we left off. So right here, make sure you had this, everyone got this right here. Squids, 10 arms. Octopuses have 8, and then the Nautilus is 80 to 90, and their eyes are way more advanced than our eyes. And I think I told you some stories of, there's been cases where an octopus or a squid will leave its tank in an aquarium and literally like escape and jump into another tank and have a midnight snack, and then go back to its tank and pretend that nothing has happened. So they are extremely successful in open water, and as a result, we have this reduction in an external shell. So they don't have like a hard outer covering like we do when we see the bivalves and the gastropods and the chitons. Nautiluses are the only ones that do. Cuttlefish and squid, they have internal shells, and you'll see that when you dissect the squid. So it does give it some structure, but it doesn't weigh it down, so it can be very fast in its environment. Octopi, they have no external or internal shell. And so how they move is through jet propulsion, where they take in water into the mantle and then they expel it out through a siphon, and you'll see that on the squid. And when they do feel threatened, they can eject water violently, shoot themselves away from the threats. They are equipped with an ink sac to cloud the predator and confuse it in the direction that it's running away from. And then there are some octopi that can camouflage. They can actually change the color, and they have pigments on their skin called chromatophores. And it does change. It's got these 3 colors, and they can change in size and diameter and texture. And then they also use pigmentation on their skin to communicate, I don't want to say emotions, but they do use it to communicate with one another. So I'm going to show you a video here. Is this the one I want? All right, bring this over.
Okay, let's play a game.
Nope, hold on here. I'm still not hooked up to my speaker. What is going on?
It's right there.
I don't know why it's not hooking up to my speaker. What do I need to do here?
Get ready.
Worked just fine yesterday. Of course not. Just got to do this when I'm recording. Don't lie to me. You're lying to me. I don't want to blow out my speakers. Well, Still not.
From Hollywood, the entertainment capital of the world.
Biologist Sarah Zielinski took these shots, and if you needed a helping hand to find the cuttlefish, don't feel bad.
I've certainly taken photos in the past, then come back to look at them and gone, I'm sure there was a cuttlefish in there somewhere.
These cephalopods are master camouflagers. But while they're hiding their body, they're revealing something about their mind, or at least their visual system.
In very simple terms, they can tell us what they can see via the body patterns they produce on their skin.
They produce these body patterns by expanding or contracting chromatophores, these little ink sacs on their skin. And they use different displays for different reasons, like for male-to-male combat.
Two males will turn into each other and pass these kind of waves of dark chromatophores over a really bright, sort of iridescent, stripy body pattern, and somehow solve these these combats. Eventually, one male, gives up and goes away.
And then there's this unsolved mystery. It changes color when it grabs a snack.
That doesn't make perfect sense because it seems to make it very conspicuous. So one theory is that, it's just a happy signal of how excited it is to have caught something, some response that it doesn't have any control over.
But most of the time, they seem to be using their chromatophores more intentionally, primarily to blend in.
Because otherwise they're more likely to be eaten, so it's very important they don't make mistakes about ambiguous visual information.
And ambiguous visual information is specifically what Zielinski's interested in. So here's the experimental setup. Print out laminated patterns, like this checkerboard, and stick them in a tank.
And we place the animals in the tank, and we record the body patterns that they produce.
You're seeing them on squares, but they do the same thing on top of circles. They produce
the disruptive pattern where you get these blocky components or high contrast components.
But when you put a cuttlefish over squiggles, it produces a
sort of mottly pattern where you get these little groups of dark spots shown across the body.
So what happens when you put a cuttlefish on something in between? When you put them on incomplete circles? When we see something like this, our visual system likes to fill in the blanks. Something we do constantly, Zielinski says.
The reason why cartoons and sketches work is because we can recognize objects based on their edges alone.
And we can identify objects even if they're broken up or
have an object that is occluded by another object. That's no problem for us. We can still work out what the object is most of the time. And I was interested to know whether cuttlefish can solve similar problems.
And Zielinski and colleagues report this week that cuttlefish do seem to
fill in those gaps and interpret those little segments as a whole circle.
Or anyway, the broken circles prompted the same camo pattern as full circles. So if you're wondering, I see these as circles too, what's the big deal? The weird thing here is that there's no reason why cuttlefish, which are
invertebrates and they're in the same group as slugs and snails,
should see the world the way we do.
Yes, it's like they're alien, but we also seem to have so much in common with them.
So, the next step?
Because we can't share the perceptive experience of a cuttlefish, it's hard to know exactly what it is that they're doing to fill in that missing information. And I want to try and get a better grasp on that and also see whether they actually respond to true illusory contours.
So you're going to show optical illusions to cuttlefish?
That's what I'm hoping to do, yes.
Okay. Hold on, I want to see if they have the other octopus video that I really do enjoy. No, okay. I don't want to read more. Show me more. All right, well, there's another one where the scientist, he's got his camera and he's just scuba diving along, and then all of a sudden it looks like a seaweed, like a clump of seaweed, and he thinks it's seaweed. And then all of a sudden the octopus turns blanche white, inks him, and propulses away, and he like screams bloody murder because he was not expecting an octopus to be there. And his partners like above, they thought he was like getting attacked by something, right? And he came up and he was just like, oh my gosh. And so then he went back under and he followed it. And then that's when we actually discovered that they could camouflage. And his video shows a way better job of them changing texture. I mean, like literally looking like coral and like blending in. I mean, like there are shots where I'm just like, I can't see it, but it's there. So yeah, cephalopods, pretty cool. Okay, slideshow. Okay, review questions. The blank of a mollusk is a highly efficient respiratory system. C. Ctenidia. The radula is the tongue, the raspin tongue. Torsion is a unique characteristic of. No. B. Gastropods. Okay, so like it's just off a little bit, and that's why they are not bilateral, true bilaterals. Elevated intelligence and complex behaviors are characteristics of the A. Cephalopods.
ND.
Oh, nice. A and D. All right. Which of the following is not a trait of mollusks? E. All choices are correct.
It's E again.
Which animal has a raspin tongue-like structure used in feeding?
C. Gotta be C. It's D.
It's actually all of them. It's all of them. E. Okay. Relate the type of food to habitat in snails, slugs, clams, and mussels.
It's not D.
That's a E?
No, I wouldn't say
C.
That's a E.
D. No, D. They dig with their feet.
Yep, they dig with their foot. They do.
So it's A.
Oh, but snails don't. They use it to push on the mucus. So it's not A. B. The foot of snails and clams are adapted for digging. Nope, that's not right. The foot of snails and slugs are flat, not allowing them to live on land. That doesn't sound right. The foot of bivalves, clams, and mussels are flat, allowing them to live on land. E. The foot of bivalves, clams, and mussels are adapted for digging. Yeah, so they dig. They bury. So it.
Not allowing them.
Yeah, well, clams and bivalves and mussels don't really live on land, though.
Oh yeah, I suppose. Wait, so.
So that would be right.
Wait a minute. So it's E. So Mary,
So it's A is what you tell me.
You know what? I'm just gonna skip this. I didn't type down the right answer, and I thought to me it seems more like E, but
That's what
yeah. All right, ribbon worms. You guys still have slides for this?
Yep.
Okay, all the way to the annelids probably.
maybe.
Purple background.
Yep.
Do we go pink?
We do review
Nope.
and then it's done.
All right. Okay, so we are still a part of the Lophotrochozoans, and so we just covered mollusca. Okay, so now we're moving on to Nemertea. I don't know. And then we'll hit annelids, and that's where we'll stop for today. So as you take a look at this, these pictures here, basically I just need you to be able to figure out similarities between ribbon worms and flatworms as well as differences. So the similarities are the ones that are bolded and underlined. So these first 3 bullet points are similarities. So they both have fine tubules that run along the body, which serves as their excretory system. They have internal organs that are not in a true body cavity. We do see some cephalization present, where we have a concentration of ganglions that kind of serve as the head, and then eye spots that do detect light. And finally, they reproduce sexually or asexually through fragmentation, just like flatworms, just like our platyhelminthines. Differences: these guys do have a complete gut. So they have a mouth and an anus. With the flatworms, we did not have that. They have a fluid-filled cavity called a rhicoceal, basically using hydraulic power and a proboscis, a very long muscular tube that can come out and capture animal prey. And then blood flows entirely in vessels. So those are the 3 big differences between ribbon worms and flatworms. Fun fact about one species of flatworms, there's one organism that was measured 60 meters in length. Okay, so to give you an idea, 40 meters would be from the first, like the pillar closest to the door to the doorway here. That's 40 meters. So all the way down by the 6th grade commons to that door, that's 40 meters. 60 meters. You got to go 20 meters beyond that.
A worm?
Yes.
Was it
Is it,
still
like,
like this
big?
big around? Was it just really skinny and long?
I think it was just super skinny and long.
Just chopped it up or what?
No, I think they're like, wow, this is a really long worm. Let's see how long it is, and they just measured it.
Use it as bait. Coil it around the hook.
Okay, annelids. So still part of the Lophotrochozoans. So the key innovation that we see with annelids is segmentation, where we have similar segments or repeated units, and segmentation has evolved multiple times. You see segmentation in arthropods, but arthropods are different from annelids, so it just arose. This is an example of convergent evolution. The advantage with segmentation is now we can have better movement or more flexibility, and then some specialized parts that could carry out either reproduction, feed-in, locomotion, respiration, and excretion. Now, annelids, this is probably the most common annelids. This is your typical earthworm. This is also an annelid. Actually, all of these are, but you could just see that it's completely different from our earthworm here. These bottom two panels are leeches in Australia. So they can be very bright and colorful, like in number 5, or, you know, dull in color, like in 6. Okay, so the annelid body has several ring-like segments, hence the segmentation, but towards one end you have the head, and we do see a well-developed cerebral ganglion. So when you open up your earthworm, you'll be able to pick out all of, basically this ganglion. You'll see it run alongside the body, and then you'll see a concentration of it towards the head. Many species do have eyes, lenses, and retinas. They have a head region and a posterior region. Each little segment is separated or divided by a septa, and they do have a closed circulatory system with a ventral nerve cord that connects all of these. So you can kind of think of all these ring-like segments as like stacks of coins on top of each other. But yeah. So another just diagram zooming in on the head. Yeah, they got a brain, they got heart, esophagus, there's that closed circulatory system. They move by contracting each little segment, so think of their body plan as a tube within a tube. And their digestive tract is straight down the center of it. Annelids move by hydrostatic skeleton. So they basically use fluid. And if you think about how hydraulics work, you force fluid into a smaller area, and then it moves levers, and you can move heavy things. And a lot of machinery that we use uses hydraulics. Well, same thing with animals here. They're using their fluids inside to move. And what they do is they have these muscles that are connected, at the dorsal to the side, down to the ventral, and they're kind of like this cross-like motion, and they contract and they push fluid as they move. And so when you see earthworms move, you can kind of see like all of that fluid bulge in, and then they just like drop, and then they kind of refill it, and then they bulge again, and they just kind of inch themselves along.
Sounds like a lot of work.
Yeah, it does. Yeah, but they're pretty fast at doing it, though. I mean, obviously, because if you drop an earthworm in the ground, if you're not quick enough, it's gone. It's already in the soil, depending on how soft the soil is. Okay, so each segment does have these tiny little bristles that help them grasp on as they bulge, and then they grasp, and then they basically move the fluid back so they can move again, called chaetea. I don't know, but bristles of chitin that anchor them as they move. So, great Australian earthworm, pretty big. Just another cartoon diagram showing earthworms. They do have a closed circulatory system. that runs along the entire length, segmented excretory system. They breathe through their body surfaces, so they'll exchange oxygen and carbon dioxide. But gases and nutrients are distributed through their blood vessels. And you'll see their excretory system in the dissection. It's very similar to the nephridia of mollusks. Now, there are some earthworms that are aquatic, and they actually have gills. So they don't breathe through their skin. They actually have gills. So kind of cool. Your book is like, annelids are composed of 2 or 3 classes. I'm like, really? You had to say 2 or 3? Just like pick one, right? So there is a current 2-class system, the Polychaeta and the clitellum. And it's where the clitellum that gets broken down into 2. The Oligochaeta, I'm butchering these names, guys, sorry. And then the Herodinia, the H1. So the clitellum, when you look at an earthworm, it looks like it's got a Band-Aid on it. That's that structure. Hold on, did I say everything? Yep. So the Polychaeta are your marine worms. Many bristles. So you can see when you look at these worms right here, there's tons of these little bristles. So that's part of the Polychaeta. Parapoda. So poda means kind of like feet, okay? But they're going to use these for paddles. So when you take a look at the cross section, 1, 2, 3, 4 of them, and they act like paddles with the little bristles at the end. Yeah, so clamworms, scale worms, lugworms, sea mice, tube worms, these are all worms. Okay, the Oligochaeta, your earthworms, it means fewer, chaetea, fewer bristles. Earthworms have between 100 and 175 segments. Mouth is on the first one and the anus is on the last segments. So when you get your earthworm, I'm actually going to make you count your segments. They eat their way through the soil. They have a strong pharynx. They have a gizzard that helps them grind up organic material. If you cut too deep in your earthworm, you hit soil because they eat soil. They deposit their waste in the form of castings. And when earthworms are actually really good for the environment. They loosen up the soil. They provide aeration. And then whatever they excrete out is actually really good for the soil. No eyes, but numerous sensitive cells. They can sense light, chemicals, because they can irritate their skin, and then touch. So I actually did a study on earthworms at Gustavus, and I was looking at earthworm density between 3 different environments. Okay, so farmland, and then I think I went prairie, and then I went river, like near a river. So in order to figure out density, this is kind of sad, but I made a meter square, and I would just randomly just toss this ring, so I didn't have like any biasness. And wherever it landed, I mixed up a solution of mustard seeds, like that had been grounded up, and water, and then I'd pour it over this meter square. And the mustard slightly irritates their skin, and they'd actually come up to breathe and just kind of get rid of this chemical. And then I just started picking them right and throw them into the buckets. Well, to figure out earthworm density, I couldn't just weigh them, because density is mass divided by volume. I couldn't just weigh them while they were moist, because that throws off your data, right?
Salt them.
No, it's something worse. Lay them out in a pan, put them in an oven,
That's awesome.
cook some earthworms. What? I don't want to, I don't know.
I
Okay?
don't know. First, let's chemically burn these worms and burn them alive.
Yeah.
What's
And then weigh them. Yeah. So
What's the most dense?
the farmland, obviously. So yeah. Rest in peace. Okay. Okay, so earthworms are hermaphroditic, but they mate. Okay, so they carry both both reproductive structures. So these are 2 earthworms maiden, and this is actually how it works. And they kind of lined up opposite to each other. And then near the clitellum is where they have the sperm vesicles. So they'll, swap sperm. And then, these cocoons will literally shed off, and these are eggs. And then they will hatch into earthworms. Yeah. So just another diagram of them shed in their cocoon, and then the young worms will hatch out of the soil. Kind of a cool thing. Yeah. Funny story. So I was doing earthworm dissections with the college bio class a couple years ago, and it rained the next day. And as I was leaving the garage, saw 2 earthworms literally mating on the pavement. I'm like, they knew I dissected their friends. And like, we got to make more. So I took a picture and then I showed it to my college bio class. I'm like,
Get a room.
okay, so the third or subclass, the leeches. So their body is flattened, almost similar to a flatworm. They are also hermaphroditic, but their clitellum, their band-aid, only develops when they are in their breeding season. They don't have the chaetea. They're not divided into segments like the earthworms are with the septas, but they do or have evolved suckers at one end that help them with locomotion and attaching to their prey. So of the species that we know of leeches, half of them eat detritus or devour small animals, and the others suck blood. And when leeches suck blood, they secrete an anticoagulant, which means that the wound doesn't clot and blood keeps flowing. And that's why leeches are sometimes used in medicine to, so like if you have, I don't want to say like an infection or something, but you know, leeches will help pump blood through to kind of push out the infection or the inflammation or whatever it may be. So it's kind of cool that they give you an anesthetic so that you can't feel them sucking your blood while it's happening.
Don't they use them to prevent clots as well? Like,
Yes, to prevent clots. Yep. But they just
put it on your body.
Yep. So just keep the blood flowing.
Wouldn't that thing be like unsanitary?
Leeches are pretty cool.
They're pretty. I don't know many leeches that carry diseases. I don't know. That's a good question. I've never thought about that. So. Yeah. I made a note here about leeches. They use leeches a lot when appendages get severed and then you reattach them. So then it says accumulations in blood causes tissues to die. So when leeches remove blood, new capillaries form in about a week. So leeches help with the development of new capillaries to those severed body parts. So here is the sophisticated bloodsucker. Here's what the bite looks like, this tri-shaped structure where anticoagulants are released. Okay, review questions. Serial segmentation is a key characteristic of which of the following phyla?
Great question.
D, annelids. Which of the following forms of annelids is not hermaphroditic?
B.
D.
Oh.
D.
They're all after.
It's always all the leeches, they're not in segments.
It's not it.
It's C. Okay. The many chaetea.
Okay.
Did I even mention that in the notes? I know I mentioned it with... Hold on here. Let me just go back. So, leeches, hermaphroditic. Earthworms. Yep. Oh, I didn't mention it, so yeah, you wouldn't know. Okay. So, the Polychaetes are not hermaphroditic. All right. Each segment of an annelid typically contains bristles of chitin called... Yeah. In annelids, which of the following chitin-based structures is used for the process of locomotion by helping it anchor while it moves? Yes, again. Chaetea. The organ is responsible for secreting cocoons in the Oligochaeta-Earthworms is known as the... Nope. What do you call the band-aid? Yep, the clitellum. The medicinal leech, once used to withdraw unhealthy blood from patients, produces blank to ensure blood flow from the host.
Anticoagulant.
Yep. Okay, so that is it for notes today.
Give us a tiny blank to put that in.
I'm sorry.
Anti-what?
Coagulant.
Can you spell it for us?
I'll just go back to the slide. Isn't it on a previous
I
slide?
wonder if I spelled it. Yeah, it was right there. It's
Right there.
Right there. Yeah.
And,
Yeah, I did.